Thank you, sir. This is Anthony Sifuna from Masinde Muliro University of Science and Technology in Kenya. So my presentation is on the need for better diagnostics for environmental pathogen detection. So you may realize that my colleagues have talked about pathogens in clinical settings, but I'll try and link this to this African situation, but in this time trying to look at the environmental situation in Africa. What we need to agree and realize is that the environment plays a very powerful role in transmission of diseases, or rather infectious diseases, within our context. Sorry, there's some interference, sir. Now, a number of environmental factors influence this spread of infections within communities in our African context. Now, one important aspect is water supply. So water supply and sanitation play a big role in this situation. And you realize that since Africa, quite a number of infrastructure are least developed, like the waste management facilities, we find that they are able to contaminate water bodies. And in the process, you find that the water the communities are using are already contaminated. So in this case, transmitting pathogens to the community or amongst the community members. Food is also an important aspect in transmission of pathogens within the environment, or as humans interact with the environment. And climate also plays an important role in these processes of transmission. So as you see, African countries or the poor countries are the ones which are most prone to climate, the elements of climate. So as we go through this climate change, so African countries are more prone to these interactions with the climate. And there are situations whereby this increase or changes in climate will lead towards increased pathogen transmission. So biodiversity, as it were, as human activities, even within the communities, these are poor communities, try to interact with the environment, getting resources from this environment, and in the process interfering with even these environments, and the process messing up with the biodiversity and the stability of these ecosystems, and the process with the people interacting with the environment, we find that these communities are quite prone to infections, and such as the case you see, cholera could be an important cause which with changes in climate can easily be transmitted among communities within the African community, African continent. Now, what are these infectious diseases that are inflicting these low and middle income countries, especially in Africa? So, as it were, generally, looking at the African situation, you find that there are numerous weaknesses within our socio-economic and political structures. And these weaknesses do provide a very good impetus to environmental factors, such that these influence a great deal of our burden of infectious disease within our populations. So, interestingly, since some of these factors are environmental, we find that the traditional health systems that are within the communities hardly manage this, and therefore diseases may be able to spread quite easily within these communities, because there are weaknesses within the environment aspect, because most of the health issues are within those traditional health systems, and therefore environmental aspects do very little in trying to manage the environmental aspects and trying to cut this connection within transmission of pathogens within communities as they interact. Now, this therefore calls for a need for strengthening surveillance within the environment, within these communities, or within Africa as a continent or nations within Africa, strengthening these capabilities in trying to strengthen our capability in surveillance and monitoring of infectious disease agents within our environment, so that we can be able to understand how this environment and the interaction between human and the environment and those different aspects can be able to minimize or cut those transmission pathways. Now, as I said, these countries generally lack poorly resourced, they lack effective infrastructure, and as my colleague Francis did present, we realize how weak even the clinical setups in trying of diagnostics are. So when you look at environmental monitoring structures in Africa, they are much weaker than even those clinical setups. So there is need for those strengthening of that, those aspects of environmental monitoring, so that these pathogens can be monitored, and then probably those controlled aspects can also be brought into action. Now, as you can see, Africa is laden with numerous pathogens that are related to environmental or are environmentally determined. So the burden would be 40%, about 40% of this disease burden is related to environmental cases, environmental factors. So you can see how important the environment would play. And if we had we developed the diagnostics tools that can be able to link between the environment and the disease transmission, surveillance, and monitoring, and it will go a great deal in trying to minimize some of this transmission and cutting of the spread of these infectious diseases within sub-Saharan Africa. So as you can see, some of these, the dengue, the cholera, chikungunya, Rift Valley fever, all these infections are important, but they are also related to environmental aspects, whether in terms of contaminated water, they could be vector transmitted. So quite a myriad of different factors which may be involved. Now, looking at the current technologies that are employed in monitoring environmental pathogens, locally or in many setups in Africa, we find that one of the most important, or which is currently in use broadly, is this culture and microscopy technique. Now, it's a good technique. In fact, in most cases, it's the gold standard confirmation methodology to be employed. But it has quite a number of limitations, as it were. So you need lab space, you need consumables, you need time to detect and make decisions. It takes time to do all this. You need personnel who are trained in trying to interpret those results, because it's more of manual interpretation of results. So you find that in African situations or in sub-Saharan Africa, you find that those are there are weaknesses in that aspect, because you need quite infrastructure and personnel to have these working for them. Now, the other aspect is techniques that can be used are indicator organisms, like this fecal indicator bacteria. So these are mostly in drinking water, agriculture, recreational. So these ones will definitely, one of the most important aspects is that although the bacterial indicators are easier to run out, but their correlation with either disease spread and also the other pathogens by pathogenic viruses or other microbes like viruses are basically not very well linked. So explaining some of the outbreaks which are related to environmental may be a bit difficult in this case if you are to use these techniques in trying to understand. Now, microbial source tracking, quite an interesting approach that has been tried in many setups, in developed countries, that is, where you are trying to relate contamination to water body contamination. But you need to develop a library or a database that you can be able to use these outcomes to be able to relate with what is happening in the environment and probably contamination within the water bodies and then the transmission of pathogens through this environment. There are other tools like the immunoassay techniques. So these are good for bacteria, viruses, that is microbes, but they are less sensitive to some extent. They are time consuming. The antibodies may not be very stable when you are, because I'll be talking about some of these environments that you are working with these kind of technologies. PCR technique is good, can be able to amplify DNA, quite sensitive, but then the conventional type of PCR has challenges in that you are not able to tell the viable versus non-viable pathogens within the environment. So although you may be able to detect this pathogen within the environment, but you see you are not able to tell is it viable so that you can be able to expand the spread, or is just the presence there, or it's not viable that it's there, but then it's not causing any disease. So you are not able to relate that with the disease. We have also this real-time PCR, which is able now to handle this problem of viable vis-a-vis non-viable detections. So this is a good tool for amplification of DNA and trying to understand. You're able to enumerate and see how if you have viable or non-viable strains because of the enumeration capability of this tool. But then the challenges would be probably false positives and also the pathogens being detected in these new pathogens which are emerging. So you find that the technique may not be able to detect in that context because you have to develop new tools or new markers for those new emerging pathogens. You have biosensors. These are also good tools, very sensitive, portable, probably very good for Africa, as it were, in the context of the situations in Africa, and as have been emphasized by, for example, Francis. But it depends with the type, actually, the type of the biosensor that you are deploying in this field. Lastly, we have the molecular detection methods, which also have been emphasized a lot with Francis. And these are my nucleic acid-based assays and DNA probes. So they are good. They are able to give epidemiological and taxonomic data, which can be very useful in epidemiological studies. So the only problem will also depend with the specificity of the genus in some species, and also strains in the different cases of these microbes. So there are those variations within the different microbes that could also influence how the level of detection and outcome for these findings, as it were. Now, why do we need these diagnostic tools in sub-Saharan Africa or in Africa as in general? Now, developing these tools to detect pathogens are quite important and will be able to help us understand quite a number of issues within our environment. Like, for example, what are the environmental drivers and determinants of these infectious diseases within the communities? Yeah, so at least we are able to understand what are drivers and what are these determinants. Again, we are able to understand vector control and disease prevention approaches that could be working, what could not be working in this case. So they will be quite good in even quality, trying to understand how our operations are working. We can also use these technologies in trying to map up geographical spread of these pathogens or disease within the environment, so that we understand these environments as and the spread of these pathogens within these environments. Now, determination of whether the intervention, as I alluded to this earlier, the intervention of these pathogens towards this pathogen spread and transmission are working. Do they need to be upscaled up? Do they need to be scaled down? Do we need to change track? So these are important things or important questions that one, as we think into trying to get into the environment and understanding how the environment and the disease pathogens transmission works or interact, I think these are pertinent questions that need answers. And as we develop tools towards this, I think that serves to make us understand better these aspects of the environment and the pathogen transmission. It also helps us sustain a successful program if it's aimed at controlling these pathogens within the environment. So if we have good diagnostic tools, then at least we can be able to understand and sustain some of these interventions that are aimed towards controlling this pathogen and the transmission. Introduction of identification of novel emerging, as you know, like now we have the COVID and many other new emerging antibiotic resistant strains emerging within our environment. So these techniques can also be very useful in trying to understand what is happening, what is new, what new pathogens, what new threats are emerging within the pathogens that could be circulating within our environment. So that's an important. And as this wastewater-based epidemiology concept developed by some is a good tool whereby by just monitoring the environment, wastewater, you're able to tell quite a number of aspects that are important within people are exposed to pathogens, they're exposed to resistant genes, they're exposed to pharmaceuticals and related other, all those aspects. You're able to monitor these in the environment, and then you can also relate them to population size and many other factors, flow rates within these wastes, the daily loads. And you're able now to anticipate, even determine prevalences, and that may be related within with this pathogen within those environments. So there is a lot of information which moving into diagnostics and we move into the environment that we can be able to dig out as we try to look for these pathogens, understand how they interact with the environment, and see the best way to control them. So lastly, what what is useful to us, or what do we need in trying to understand or to be able to understand the environment and pathogen relationships within our context here in Africa. So there are these emerging developments that may be useful for us as we try to develop tools towards detection of pathogens in our environment. So one could be these aspects of new developments in electronic surveillance systems, where information is related and transmitted within different aspects of from the source to probably a national control center somewhere which does put together the data and try. So you need tools that are able to or are amenable to such like new developments, new developments which can be able to link, use cloud, use telephone, mobile telephone technology, so that it transmits these results to central centers where this information can then be interpreted and then be useful or used by governments. So one of such a technique or tool that is being developed by the Imperial College, this is the Less-Wing digital platform. I think can serve very good when currently is in use in clinical diagnostics, but it can also be tried out in environmental situations, and we try to see how it works. So this would allow for rapid problem identification and also action being taken up. So this tool also needs, these tools need to be rapid pathogen detection methods, rapid turnaround within, for example, 30 minutes, within 25 minutes, such that results can come out and actions can be taken up very fast. We are talking of fields in Africa which are remote. There is a lot of dust. So these field situations where you get a tool, get to next to the river, get to the forests somewhere and try to get samples, analyze them in the field, put them onto mobile phone and transmit them to a central point somewhere. And this will try to solve quite a number of issues that or challenges that are present in Africa. So deployment of low technological environments. We are looking at a situation whereby you don't have laboratories within the field. You don't have reliable electricity. So we need technologies that can be may not require these complex laboratory buildings, do not necessarily need electricity. Probably you can be able to connect them to solar powered technologies, and in the long run end up with getting running the machines, and then you're able to generate the information. Exploit the data, that is bioinformatics, which is a field that is very useful now, very good in terms of tracking disease outbreaks, and also virulence or pathogenicity of these diseases. So in linking these technologies with such like emerging fields like bioinformatics would really add great value to these new technologies. Now, lastly is the aspect of costs. So we need cheap but reliable, cheap but reliable detection technologies that can be used in a remote and rural setup. So this basically what I use for reference, and then I'm quite grateful to the Digital Africa Digital Diagnostic for Africa Network, which brings together we as scientists in Africa and the world generally to try and see how we can be able to develop tools that can be useful for the next generation of tools that can be useful for Africa. I'm grateful also for having been hosted on this conference. Thank you.